Good afternoon, everyone. Welcome to the ANRF Pair Project webinar. It is my pleasure to introduce Dr. Usha Rajamma, Senior Scientist, Mentor and Researcher, whose career has spanned more than 3 decades in biomedical research, education, and translation research. Welcome, ma'am, to the webinar.
Thank you for the introduction. And screen window share. Hello, the slide is the slide visible?
No, ma'am. Now it is visible, ma'am.
Okay. Hello, welcome everyone. And I'm working as a scientist at Inter-University Center for Biomedical Research and Super Specialty Hospital. This is our institute. It's a Kerala government funded institute under the mentorship of MG University, and I am going to present our part of the project in ANRF pair project, epidemiological study, biomarker discovery and drug development for diabetes. So as we all know, diabetes is a global health challenge, and this condition is characterized with a high blood glucose level. Am I audible?
Yes, ma'am.
Yes, ma'am. And there are basically different types of diabetes, and two types which are very common, that is type 1 diabetes and type 2 diabetes. Among this, type 2 diabetes is highly prevalent in almost 90% of the diabetic cases, and type 1 diabetes is around 5 to 10% among the total diabetic population. So how these are either insulin dependent or insulin independent. So in the normal case, the pancreas produces insulin, and it binds to the insulin receptors in the fat or adipose cells or any cells, and where the insulin receptors are present, and it elicits a signal transduction, and that signal transduction causes the glutamate transporter to move to the surface, and the glucose will bind to this glucose transporter, and it is taken inside, where the glucose is converted to triglycerides in the cells, and it produces energy, glycolytic reactions also. So this, when in type 1 diabetes, what happens is the insulin is not synthesized due to the loss of beta cells. In pancreas, beta cells of islets of Langerhans produces the insulin, and so the lack of insulin production prevents the glucose to bind to the glucose to be transported into the cell. So the amount of glucose in the circulation remains high. And in type 2 diabetes, there is enough of insulin in the system body, but the cells do not use insulin because those receptors are insulin insensitive. Therefore, the glucose level in the blood goes high and it cannot be transferred to the cells. So there are other diabetes types are also there, that is gestational diabetes, and that develops during pregnancy and goes away just after childbirth, and there is a high chance to, these people have high chance to develop type 2 diabetes. And another case, the fourth case is pre-diabetic cases. That means there is a high blood sugar, but there is the sugar levels will be more than the controls, but not enough to diagnose as a type 2 diabetes. Okay. And I am audible, isn't it? You can hear me?
Yes, yes.
And the fifth case is monogenic diabetes. That is, diabetes is developed as a result of a genetic defect in a single gene. That can be, there are some genes like defects are detected in glucokinase, hepatocyte, nuclear factor 1 alpha, and 4 alpha. These genes, these defects have been observed, and they causes the monogenic diabetes. And coming to the difference between type 1 and type 2 diabetes, in type 1 diabetes, body will not be able to make enough insulin, and whereas in type 2 diabetes, insulin is produced, but the body cannot respond to the insulin. And type 1 diabetes is caused by immune system, it's an autoimmune disease, and it starts in childhood. But the other type 2 diabetes is caused by lifestyle changes, and genetics also contribute. So it is a complex disorder, and it is more common among middle-aged people. And this type 1 diabetes, the symptoms occur very quickly, and it is treated with, it can be treated with insulin injection. It is treated with. But in type 2 diabetes, the symptoms develops very slowly, and it is managed with drugs and lifestyle changes in initial cases. And the medications which is used for diabetes are for managing the blood glucose levels, and most of the drugs act through different mechanisms. For example, metformin is a common drug given for type 2 diabetes, and which decreases the amount of glucose produced via hepatic gluconeogenesis. Gluconeogenesis means, the hepatic gluconeogenesis means the glucose production in the liver that occurs when and its precursor is non-carbohydrate sources. So like amino acid, pyruvate, and glycerol. And this drug improves the insulin sensitivity also. But another drug is glimepiride that stimulates insulin production by the pancreas independent of blood sugar levels. So it always in stimulating this can be combination drugs, or it can be given singular also. And the third type of common drug given for diabetes is pioglitazone. It activates the insulin receptors that regulates the genes related to glucose transport, which helps reduce the insulin resistance. And the fourth type of drugs like empagliflozin, dapagliflozin, that actually protects the kidneys, because it blocks the glucose reabsorption in the kidneys and helping the sugar to be excreted through the urine. So it's not building up in the kidneys. And as I told you, diabetes is it's a growing global and local health burden. So, and it is driven by aging population, obesity, and urbanization. And of course, there comes the lifestyle changes. And over 90% of cases are type 2 diabetes, I told you in the beginning. And India is among the most heavily affected nation. And if almost 11 to 16% of the total population is affected with diabetes. So why do we need an epidemiological study for the diseases? It will give us an idea about the distribution or prevalence of the disease, and it will identify the determinants of the disease. That means causative factors or what are risk factors, and the complications across the population. So a recent the means IDF, International Diabetes Forum, they according to the recent estimate, about 580 million people, adults, are living with diabetes worldwide, and that is 90% of the globally identified cases of diabetes, type 2 diabetes. And India, there are around 101 million diabetics people are there in India, and 136 million pre-diabetics are also living in India. So the tracking the distribution, determinants, and complications are necessary. There are in epidemiological studies, we get all the information in 5 W's. So what are the 5? What is the what means? It's a clinical condition. The type, how clinical condition means which type of disease, what is the severity, and how it is affected, how much of population is affected, and who the person, the demographic information on the people who are affected by the disease, and how it is, where it is affected, which means whether it has happened when they were in the workplace or in other cities, or it has occurred in their own hometown, and the time over a period of time, how it is developed. And that is called descriptive epidemiology. These informations will give you the descriptive epidemiology. And then the second case is analytical epidemiology, where you can find why and how this happened. So the causes, risk factors, and modes of transmission will be collected. These informations we will get through the analytical epidemiology. So this will lead to the hypothesis formation for the causative and risk factors for the diseases. And there are so many complications occurs due to the diabetes. That includes peripheral neuropathy, stroke, retinopathy, neuropathy, peripheral arterial diseases. If untreated, it will cause so many other multi-organ complications. So it is better to have a knowledge in a particular area, how much of people are affected with the disease, what are the steps to be needed, is whether it is going into an epidemiology, means epidemic sort of condition. We will be through the epidemiology studies. So what measure, what is the test we can do for screening the population for diabetes? One is fasting blood glucose, and the 2 hour after food, that postprandial glucose level, and glycated hemoglobin. And glycated hemoglobin test, the HbA1c test, is the gold standard for measuring the blood glucose level. Why is it so? Why the other 2 are not that good? Because that blood glucose level, usually it affects the daily blood glucose level, that fluctuate constantly based on what we eat and the amount of stress what we are undergoing, or the degree of physical exercises we are doing. So HbA1c reflects an average blood sugar levels for the past 2 to 3 months. So it's a average blood sugar for the last 2 to 3 months. So the progression of and how it is maintained, the sugar level is maintained throughout this period, we will have an account through the HbA1c measurement. So HbA1c measurement gives us a long-term picture of blood sugar management. So it is good for screening, diagnosis, and prognosis of the disease. Glucose, what are we measuring in HbA1c? Hemoglobin is the protein carrying oxygen in RBCs. And this hemoglobin is getting glycated or conjugated with the glucose moiety. And RBC lifespan is around 120 days. So this any changes in hemoglobin level is an average, HbA1c is an average of this many days. So the glucose level attached to hemoglobin A1 subunit represent the blood sugar history during this period. So that is why this is a gold standard for measuring the glucose level in a diabetic patient. And what is the existing lacuna? Because there are few studies. There is a major large-scale survey has been done by ICMR, India Diabetes Program. So that gives the diabetic prevalence in Kerala as high as 24%. But this is suffering from some problems. So what are these? They have used around 1,24,000 people, and that is from 31 sites, I think, 31 sites. So there is an aggregation bias, and it masks the low-level heterogeneity. And there is an underrepresentation of marginalized pockets. Usually it was done. It has actually Dr. Mohan from Chennai. He was also involved in this investigation. He was leading, in fact, I think. And there is a large percentage of cases where from the urban areas, and this rural and semi-urban parts of Kerala and other states were remaining undiagnosed. And it relied on several data and self-reporting systems. So that to overcome that, we thought that there should be a localized census-based cross-sectional studies in a particular area. We have taken a panchayat to resolve these gaps so that it captures the granular, community-specific data, and it will detect the undiagnosed cases of diabetes. So the rationale for undertaking this sort of study, a cross-sectional study, is to overcome these gaps, and we proposed a ward-by-ward cross-sectional study in a multi-ward panchayat of Pudupalli. So, and it will result in a universal screening with total population coverage irrespective of the age group, and it eliminates sampling bias because it is all-inclusive epidemiology study, and we use standardized clinical methods, and it will micro map the environmental risk factors, and of course it can provide an actionable registry data for the local panchayat as well as for the public health centers and ASHA workers. That will help in targeted public health interventions and to map the disease clustering in different wards and exact prevalence and risk factors associated with the disease, and it will monitor the disease progression. So our objective is to develop a community-based diabetic surveillance framework for diabetes to map diabetes and pre-diabetes prevalence, to identify the high-risk groups, to identify the complications associated with diabetes, and to stratify diabetes by stratify the risk of diabetes by the age, gender, socioeconomic status, and lifestyle factors, and to suggest, and it will help in suggesting targeted preventive interventions across the region. So the workflow is like this. We have selected a target population, And the geographic area, it is the general population from the 19 wards of Padupalli Panchayat. And the indicators include prevalence of diabetes, pre-diabetes, risk factors, and complications of diabetes. Okay. And we have engaged stakeholders like we have got ethical clearance, local panchayat officials, we have obtained the approval, and we have engaged local ward members. They provided the census-based list of households and engaged public health center clinicians in the study. So this much of part of the workflow has been completed, and we are proceeding towards the recruitment of and training of the field workers, like engaging ASHA workers, community workers, and researchers and volunteers with questionnaire for screening. And then we will go for the preparation of database depending upon the information we are collecting. And then afterwards, we will arrange medical camps at the local public health center to confirm for the confirmation and identifying high risk cases for primary care and treatment and follow up. Then we will analyze the data to get the prevalence estimate, distribution, and risk dashboards, and share these findings with the local panchayat and the clinicians. And we will suggest, this study will suggest targeted intervention to tailor diet, activity, and awareness campaigns to identify high risk groups. And we will review the quarterly performance indicators and then refine protocols as and when required. And we will, if needed, we will secure funding for continuity. And we are suggesting a slight modification in the methodology, because this part of this, duration of this proposal is for 2 years. We have completed an earlier epidemiological study on neurological disorders during 2018 to 2021. And this is the paper published from the epidemiological study. And in that was also done in Pudupalli Panchayat. And it was an all-inclusive study, screening study for neurological disorders. It included 6,771 households with 26,465. It covered 65 individuals from newborn to adult around 106 years. So in that questionnaire, we had a question in that questionnaire, which is about, does any member in your family have any of the following diseases? In which we have included diabetes also. So we have a data of diabetic people in this panchayat during the 2018 to 2019-21, and we will approach them with the questionnaire. And we will also we have got the contact details of all the people in this panchayat. So we will, their contact numbers are also there. So we will contact them with the questionnaire and try to find out the new cases which has come up, and they will be invited for the medical camp at probably in the CHC of IUCBR and SSH. So the societal part is the first part, and the second part is the laboratory work. So we are towards biomarker discovery. So the patients identified through the epidemiology survey, medical camp, the controls as well as the cases, diabetic cases, and through the community health center, we will recruit 15 controls, 15 pre-diabetic and the 15 diabetic cohort for blood sampling. And serum will be, from the blood sample, serum will be used for proteomic analysis. Whole blood RNA will be used for transcriptome, means gene expression transcriptome analysis. And with this data, we will be able to identify the differentially expressed protein, we will be going for the targeted DNA sequencing for any variants associated with the genes which are identified through the targeted DNA sequencing. So this way we are getting the differentially expressed protein and RNA, but this portion was not introduced in the proposal, So we will, based on the other two, we will go for the differentially expressed protein and our proteins and genes, and then confirmation of this in this differentially expressed protein in more samples. At least pick up some of these which are having a high score of differential expression. We will take those samples for and confirm it in more samples through RT-PCR, specific RT-PCR, Western blot, or ELISA if antibodies are available in the market. And then finally, it will lead us to identify the biomarkers and drug targets. So this part of the work, maybe we are trying to finish it by the third year, and then go for when the targets we identified, we will do an in silico screening of anti-diabetic molecules from Ayurvedic herbal sources, utilizing the novel targets identified from transcriptomic and proteomic analysis. And then we will do some in silico studies to find out the pharmacokinetics and toxicity, and then we will go for the in vitro screening. And these parts, most of these parts, Dr. Gautam Chandra will be leading this work. And then we will go for the in vitro screening in cellular models to assess the glucose uptake, insulin signaling, and lipid metabolism, and mechanistic studies to investigate molecular pathways influenced by the bioactive compounds. And the in silico screening and computational work will be carried out by Dr. Deepthi Varghese, and mechanistic studies on including ANPK activation, glutamate transporter translocation, and modulation of oxidative stress and inflammatory markers. This will be studied, and then these will be in vivo validated. These promising candidates will be validated in high-fat diet-induced genetically modified diabetic rodent models, and then toxicity determination of safety profile, bioavailability, pharmacokinetics, therapeutic applications. This is what our proposal is, and this particular where the data and the idea was provided by all these Dr. K.P. Mohanakumar, Dr. Gautam Chandra, Dr. Rajesh Shernai, Dr. Deepthi Varghese, Dr. Sitama Sen Chandra, Dr. Mridula Matthew, Dr. Emya VR, Dr. Emya VR, Dr. Rakesh, and Dr. Niranjana Gopal. They are the Ayurvedic doctors. And Sito Nelson is the project associate working in the ANRF prayer project. And other co-investigators include collaborator from Professor Anil Kumar from he's the collaborator from University of Hyderabad, the Hub Institution, and Dr. Sadi. She is on drug discovery project. So she is the collaborator from MGU, and I thank Dr. Radhakrishnan, the NRF Payer Project PI, and thank you.